So here we can see this process. We see ATP is a part of the process. The large purple thing is the ribosome. One thing that we do need to note in AP biology is that the ribosome is really made up of two parts. There is the small subunit. Here, excuse me. Here it's just kind of referred to as 40S. That's just a reference to the size. And the larger subunit is referred to as 60S, another reference to the size. Oops. So let's take a look at this very first part. So in step one, this protein RNA complex, which is called the small ribosomal subunit, recognizes that 5' GTP cap that we talked about last week. And in doing so, it binds to the very start of this mRNA transcript. We call it a protein RNA complex because it is made up of both. There are some proteins, but it also is made of RNA as well. So don't be confused about that. And more frequently, we will just refer to it as the small ribosomal subunit. Part of that complex includes what's called the MET tRNA. And it's called the MET tRNA because MET is an amino acid, methionine or methionine. And methionine is going to be the very first amino acid. This tRNA recognizes the start codon, AUG. And because of this, it brings with it the very first amino acid. So I'm going to go back really quickly. I will come back to this slide. So if we're looking here, we can see this codon, AUG. It is almost always going to be the start of translation. That's why we call it the start codon. And its corresponding amino acid is MET, methionine. Now, notice that, we talked about this on Friday as well. In most other cases, several different codons will code for a single amino acid. And we can see that looking at this chart. But in the case of methionine, there's only one codon that codes for it, AUG. Let me know when you've written down whatever you want to write down here.
Okay.
Okay, and then the second part, we can see that there is the involvement of ATP energy. And using energy allows the mRNA to move through the ribosome. And of course, its 5' end is moving first. So it's kind of like, if you go head first down a slide, the 5' end would be the head. It's going first through this protein complex. And this is going to continue until the ribosomal small or the small ribosomal subunit finds that start codon, and then it kind of gets locked in.
I'm done.
And the last step here of translation, now we can see that the larger ribosome subunit is going to come and attach to that smaller subunit. Now we're going to actually begin this next step called elongation, which is when the mRNA is going to move through the ribosome. So really the last part of initiation is just this attachment of the large ribosomal subunit.
I'm done.
Okay, now elongation. Elongation is a somewhat more complicated process, but I think I really want you to pay attention to this image here, and hopefully it'll make more sense. There are some intricate details here, though. So the first part of elongation is to actually recognize the codon. And a codon, remember, is a group of 3 nitrogen bases on a transcript of mRNA. So the ribosome is going to just keep scanning for the next codon. And whatever message that codon is carrying, whether it be AUG or AUC or CCG, whatever the codon is, there's going to be a tRNA with the corresponding anticodon that's going to move into what's called the A site of the ribosome. So if we look at this image, we can see there's an E site, there is a P site, and then there's an A site. I can't remember what these actually stand for, but I've always kind of thought about them as E standing for exit. This is where the tRNA is actually going to leave the ribosome, as we will see. P stands for protein, because as you can see, the protein, the new polypeptide, is kind of growing out of this P site. And then A stands for arrival. So the new tRNA is going to move into the A site. Now, of course, again, like I said, that's not actually what it stands for. I'm pretty sure it's a more complicated chemical phrase, but this is how I've always remembered it. And again, we have to, and you can see it in this image, we have to use some GTP, some guanosine triphosphate, as a source of energy here to help the process be more accurate.
I'm done.
Okay, so now I want to ask a question. We're talking about here corresponding anticodons. The anticodon is like the bottom section of a tRNA. You can kind of see it here. You can see those 3 small, those would also be nitrogen bases. So let me ask you this. If I've got a codon that is GAU, what would the anticodon be for that?
CUA.
Excellent. Okay. Just wanted to make sure you were clear on that. The second part of elongation is called the peptide bond formation. So the ribosome is actually going to form peptide bonds between these amino acids, contributing to a growing polypeptide chain. So the polypeptide chain is growing. In the P site, what's going to happen is that the ribosome is going to form a new peptide bond between the chain and the new amino acid that's in the A site. So EPA, exit, polypeptide, and arrival. Oh, I feel that.
I'm done.
And then the last part of elongation is translocation. You don't need to remember the exact names of these steps. You just kind of need to know what's happening throughout this process of translation. So, peptide bond formation, translocation, these phrases are basically just made up. But as long as you know what's happening in the process, you're good. So the tRNA that was in the A site is going to move into the P site. And the tRNA that was in the P site moves into the E site. So we've got this right to left movement. The tRNA in the E site is then going to be released. So that's why I always think that the E stands for exit. Of course, it really doesn't, but that's kind of how I remember it. And as we can see at the very bottom of this image, this also requires energy in the form of GTB.
I'm done.
So termination of translation. I think this is the only slide I have for this. Yeah. Termination of translation happens when that elongation continues until it reaches a stop codon. So there are three possible stop codons: UAG, UAA, and UGA. When the ribosome comes across one of those codons, that means that it's time for the translation process to end. What's going to happen is that what's called a release factor, which is shaped similarly to a tRNA, is going to enter the A site. that triggers hydrolysis, and the growing polypeptide chain is going to be literally cut off from that last tRNA. And as it's cut off, it starts to float away from the ribosome. It's now considered a free polypeptide. And the ribosome is going to disassemble. The large subunit is going to move away from the large subunit.
Okay. I'm done. But where is the definition? Is the definition for the release factor that it hydrolyzes the final amino acid?
Yes. Yeah.
Okay. Hold
Good.
on. Change it.
And you can also include that it's shaped similarly to a tRNA. That's what allows it to actually fit into the A site.
Okay. Okay, I'm done.
All right. So essentially, that's what I wanted to go over today. One more thing that I want to mention is that as this polypeptide is released, it becomes the primary shape of a protein. Do you remember anything about what that means? Primary, secondary structure, tertiary, quaternary structure. Do you remember that at all?
Yes, I remember that. I just don't remember what they look like. But I remember that from like one of our student study guides, or was it like a reading we did?
Okay. Yeah. So keep that in mind. We'll talk more about that in our next lesson. But we have to be careful with our language here. And there's a reason why this says polypeptide and it does not say protein, because a polypeptide is just the primary structure of what could eventually become a protein. But in order for that to happen, there's going to need to be a complex folding process that needs to take place. So this is literally just a chain of amino acids. Eventually, it could become a protein, but that is a slightly different process, and we'll talk more about that in our next lesson.
Okay.
Yeah, I don't want to get too far ahead right now because Selena isn't here, but this is the process. Do you have any questions about any of these 3 steps: initiation, elongation, or termination?
No.
All right. Okay. Well, I know you're probably looking forward to getting some more rest, and I need to do some stuff for my bio class. One thing I do want to mention is that March is National Women's History Month, so we will kind of focus a little bit on some of that history. But primarily, I just want to say that I'm grateful for you, and I think you've been a great student since I met you a couple years ago, and I'm excited for what's to come. And I really appreciate the patience that you've shown me, and you've taught me a lot about what it means to be a good teacher. So I appreciate you, and Yeah, I look forward to the remaining time we have because graduation is not too far away.
Yes. Thank you, Mr. Red.
You are welcome. All right, I will talk to you tomorrow.
Okay. Bye.
Bye-bye.